Hokey is a puppy of a breed that I would refer to as a border collie, and you guys would probably refer to as a heading dog. Hokey has got a genetic disability, and that's not a surprise looking at her, because the genetic disability that we're talking about has two forms. One of the forms is linked to the gene for white colour coat, and the other one is linked to the gene for merle colour coat, this kind of livery brown colour that she's got on her head here. Hokey is deaf, and she's deaf because of a condition known as congenital deafness, which we see in various breeds of dog. Not surprisingly, the dogs with predominantly white coats or large amounts of white coat. We see it in Dalmatians, we see it in English Bull Terriers, we see it in Border Collies, we see it in a list of about 20, 25 different breeds of dogs, mostly associated with that white coat colour. And we also see it in cats with the same associations, so that British cats are more affected than other breeds of cats, generally speaking. I'm going to start off talking about the cochlea and talking about the neurophysiology of the transduction of pressure waves into sound information and how that's dealt with in the central nervous system. So let's talk about the hair cells to start with. This is a rather pretty, I think, scanning electron micrograph of the hair cells in the cochlea. And the business end of a hair cell are these so-called hairs, and each array of hairs here is attached to one individual hair cell. Those hair cells, or the hairs on the hair cells, are actually specialized cilia or flagella-like appendages, and they are movement transducers. We've seen a light transducer in the last lecture. We'll talk about transducers in a more general sense when we talk about the sensory systems next semester. These transducers are movement transducers. They change the membrane potential of the hair cell in response to a movement of those cilia. Let's see how they do that. If you look here, we have got a transducer which doesn't generate action potentials, and a sensory nerve which does generate action potentials, just like the retina, although not quite as complex as the retina. So, if something moves the hairs in this direction, we get a depolarization, and if something moves the hairs in this direction, we get a hyperpolarization. And that depolarization and hyperpolarization increases the rate of action potential generation from a resting rate, or decreases the rate of action potential generation from a resting rate. Just like the ganglion cells in vision, and the standard model for a transducer sensory neuron interaction. The interesting thing about the hair cells is that they are a more normal sensory transducer, in the sense that they are less specialized and more flexible. If you think about the visual transducers, we only find them in the eye. They're extremely specialized, and they respond to light in the eye, and their only job is to put together the visual image, or to begin to put together the visual image. These transducers are much more flexible and their function depends on where we put them. So for this morning we'll be talking about these with reference to hearing, but next semester we'll be talking about those same sensory transducers with reference to the sense of balance. Same transducers, we put them in a different place, we wire them up slightly differently, and they respond to different things. So in terms of the sense of hearing, how are those transducers wired up in order to allow us to hear? This is a tympanic membrane. In fact, it's a whole cochlea. And what's been done here is it's been unwound. The cochlea is kind of spiraled around itself, but functionally it's easier to understand if you stretch it out and consider it as a straight thing. It's just that it doesn't particularly fit into a skull quite so well if you stretch it out, because it needs a long, thin cavity and it will be more damageable and more fragile. The structures that convey the sound impulses across the middle ear cavity, the three ossicles, abut against the oval window, and effectively there is a fluid-filled cavity which goes all the way up and all the way down the cochlea, and it gets back to the middle ear cavity here with a round window. The hair cells are embedded in the organ of Corti, and that is this structure in the middle that's labelled as basilar membrane. The basilar membrane is part of the organ of Corti. What we have there is we have a membrane which is strung across these effectively two fluid-filled spaces. It's strung across tightly, and the hair cells are embedded into it at one side. The width and tautness of that membrane varies along the cochlea. So, if we imagine a positive pressure wave coming and hitting the tympanic membrane, being transmitted across the middle ear and pushing in on the oval window, as it travels through this fluid space, the pressure wave distorts the organ of Corti. It distorts the basilar membrane. And conversely, as a negative pressure wave is transmitted through the middle ear, the basilar membrane is distorted in the other direction. You'll know from physics that sound is a pressure wave vibration that's set up in the atmosphere and transmitted through the atmosphere to the ear, and so you need to imagine that this is vibrating in and out the whole time in response to sound, and that this membrane is moving up and down. And because the hair cells are embedded in the membrane, the cilia, the hairs, are moving backwards and forwards and altering the membrane potential of the hair cell. Because the length and tension of the basilar membrane is different at different points along the cochlea, that means that different points along the cochlea have different resonant frequencies. And again, from physics, a resonant frequency is the frequency at which that structure is preferentially able to absorb the energy from a waveform, preferentially able to vibrate with the waveform. And an example of a thing with a resonant frequency, well, everything has a resonant frequency, but an example of a thing that you'd be familiar with a resonant frequency is a tuning fork. Yeah? You take a tuning fork, hit it on a table or surface, you put it on the surface, and it sings out a note at its resonant frequency. Yeah? So structures have a resonant frequency. The basilar membrane is no different, except that because its width and tautness varies along its length, its resonant frequency also varies along its length. So if we give it sound at a particular frequency, one particular part of that basilar membrane will resonate with that sound at that frequency. And the hair cells that are embedded in the basilar membrane at that point will move up and down more than the other hair cells. And the neurons that are attached to those hair cells will alter their rate of generation of action potentials more than the other neurons. And we will go, middle C. There is an order of frequency response across the cochlea. So that low frequencies tend to be resonating and so sensed near the tip, and higher frequencies tend to be resonating near the base. And you will probably know that as you get older, you lose the ability to hear at the very edges of your cochlea, particularly the high frequency edge. So that when you're a bright young thing like yourselves, you can probably hear to around about 23 kilohertz, 23,000 hertz. When you're getting old and a bit doddery like me, it's dropping to around about 20 or 19 kilohertz, I should hope. And as you get older, your high frequency sound drops off. And you'll probably also be aware that if you have a history of attendance at Black Sabbath concerts in your youth, your high frequency sound will tend to drop off in later life more than other people's. Different points along the cochlea respond to different frequencies of sound. And if we hear a complex sound, let's imagine a triad chord, a sound that is made up of 3 different frequencies at the same time, we get 3 different areas of response. Every sound is made up of a characteristic set of frequency components. And so by separating out these frequency components, we can interrogate the sound that we're hearing and characterize it and work out what's going on with it. Interestingly, in the same way that signal engineers do when they're analyzing waveforms electronically. So, there's a physical change in a cochlear organ transduced to a membrane potential change by a hair cell, coded as a change of rate of action potential generation by a neuron, and now being sent up to the central nervous system for processing. What we now need to look at is how this information travels up the central nervous system. Another example of hierarchical processing, as you'll see. And also, I want to highlight the way the information is changed as it goes up the central nervous system. Because what happens as information goes up the central nervous system, we talked, second lecture I think, about whether or not information is dealt with locally or whether or not it's passed up to a higher level of the central nervous system. Remember the dead mouse at McDonald's? What we also need to appreciate is that if information is passed up to the higher level, more processing is done on it as it goes up, and the information derived from that processing is passed on as a kind of added value to the information. So here we have the centers in the brainstem and the cortex that are responsible for dealing with the sensation of sound. Sound is processed at all these places in the brainstem as far as the medial geniculate nuclei and the associative cortex and the primary auditory cortex. We'll see that in a moment. And each of those places has something new to add to the information that we get with the sound. So, for example, as we'll see in a moment, there are cells in the cranial olivary nuclei that work out whereabouts in space that sound came from. And then they send that sound information onwards and upwards with a little added tag that says, came from over there. The colliculi and the geniculate nuclei play similar roles to the roles that they play with vision. So it won't be a surprise to know that the caudal colliculus, rather than the rostral colliculus for vision, is the place where lots of information is sent to lots of other systems around the body. To the postural system, to the reticular formation, to various other places. Nor should it be a surprise that the medial geniculate nuclei are the places that send the information onto the cerebral cortices. But notice that there is processing at every level, and there's also crosstalk or decussation at many levels within this pathway. Just to convince you that this is a real pathway, here is the dorsal view of the brainstem of a dog. And I've taken the cerebral cortices off, and the cerebral peduncles, the attachment of the cerebral cortices, are where the blue dotted lines are at the top. And I've also taken the cerebellum off, and the cerebellar peduncles are where the red dotted lines are. So we've just got the brainstem, with all the brainstem structures that we should be familiar with, the medulla, the pons. These two round things here are the colliculi. This is the rostral colliculus, this is the caudal colliculus, here's the medial geniculate nucleus. All the structures that we have looked at in this diagram are real structures that exist on a real brainstem or in a real brainstem. Travelling further upwards, and this time a horse's brain, just for variety I suppose, and to kind of prove that horses do have a brain. The primary auditory cortex is very lateral here on the temporal lobe and also on the other side. That's where the primary auditory cortex is within the cerebral cortices, the first place within the cerebral cortices that responds to an auditory stimulus. And that gives me a chance to talk about another physiological principle that we haven't discussed yet, called somatotopic localization. Somato of the body, topic, placement, localization. The body placement is localized on the brain. We see that with all of the neural pathways. We see it at all levels within the neural pathways. But here's an example of it in the primary auditory cortex. So what do I mean by somatotopic localization? Well, what I mean is that the response to sound on the cochlea is a spatial thing. Yet the frequency of sound response changes as you move along the cochlea. And in exactly the same way, the frequency of sound response changes as you move across the surface of the primary auditory cortex. So at one side of the primary auditory cortex, we're dealing with low frequency sounds, and as you move across to the other side, the sound frequencies increase and increase and increase until when you get to the other end, you're dealing with high frequency sounds. Just the same as in the cochlea. The arrangement of the auditory cortex is the same spatially as the arrangement of the cochlea. Somatotopic localization. What I would like to do now is to talk about the electrical activity of this sound system and talk about how that processing alters our perception of reality. So back to the brainstem. If we measure the electrical activity of the brainstem in response to a sound stimulus, there are several things to say. First of all, it's pretty rapid. this bar represents 1 millisecond. And secondly, it's very small. This bar represents 1 microvolt. It's so small, in fact, that it is lost in the background activity of the electroencephalogram, which typically from a dog is of the order of about 4 to 10 millivolts. So we're trying to find a 4 to 5 microvolt signal in 4 to 10 millivolts of background noise. Not going to happen. The nice thing is that the electrical response to a sound stimulus is exactly the same every time. But the background noise, the electroencephalogram, is random. Well, we don't actually believe it's random. We believe that the electrical activity of the brain relates to the function of the brain. But the function of the brain is so complex that it's effectively random. It's so complicated that we can't predict what's going to happen to that overall electrical change from one moment to the next. And so we consider it to be random. If you take a lot of random waveforms successively and add them all together, you end up with a flat line. Does that make sense to people? Let's draw that a little bit of a complicated idea. If I have got a random waveform that looks like that, and another random waveform that looks like that, and a random waveform that looks like that, and I add all of those together and divide by the number of waveforms, the average of all those waveforms is a flat line. And the more times I do it, the flatter the line gets. Yeah? The less the error gets in the experimentation, and the flatter and flatter it gets. So, if I expose a dog to a click stimulus and record the electrical activity that follows from that stimulus, most of that is random, because it's EEG from areas of the brain that are wondering when the next dinner's coming along and wondering where they're going to go for a walk that day and all that sort of thing. But some of it, a very small part of it, is going to be the electrical response to that click stimulus. And that part, that very small part, is going to be the same every time. So now, if we add together sections of electroencephalogram from immediately after that stimulus, we don't get something that tends to a flat line, although the random part of it does tend to a flat line. As the random part tends to a flat line, we're left with this, which is the response to the click stimulus. And so it's possible from an adult awake dog to record the electrical response of the brain to a click stimulus by giving it, say, 500 click stimuli at 10 a second and averaging the response to each one until we get this. And it's been shown, not in the dog this time, but in the cat and in the monkey, species that tend to have brains that are quite amenable to research, it's been shown that these waveforms come from specific nuclei within the central nervous system. So that the cochlear nucleus is wave 1, the auditory nucleus is wave 3, the caudal colliculus is wave 5. We don't know where all the waveforms come from. We're not sure at the moment where waveform 4 comes from, but we've got an idea of which centres in the brain are generating which of those waveforms. And so that gives us a very important concept, the concept of delay in processing. Because the caudal colliculus can't get to the sound until all the other nuclei between the ear and the caudal colliculus have dealt with it, processed it, passed it on. So as you go up the central nervous system, the delay following the stimulus gets longer and longer. Going back to the horse's brain, because this is an auditory evoked potential, that's what we call those things. This is an auditory evoked potential recorded from a pony. Now we're getting further out, we're getting longer. So this is now the microvolt, but this is now 10 milliseconds. So we're looking later on in the response. And we are seeing here a response from the primary auditory cortex. Here is a thing called a post-auricular response. That's the nervous response you get as the horse twitches its ears towards the direction of the sound. So that comes from the external auditory muscles. And here is a response from the associative cortex. The response from the primary auditory cortex in a horse occurs at about 12 or 15 milliseconds. The response of the associative cortex occurs at between about 30 and 90 milliseconds. And much later on, this time demonstrated in humans, is a conscious or associative response. We call it the oddball response for reasons that will become apparent in a minute. That is the first response that can differentiate between a sound you're listening for and a sound you're not listening for. So if we take a human volunteer or patient, sit them in a room, and we say, we're going to play you a series of clicks, and every so often there'll be a bleep, and then we go click, 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 bleep, click, 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 bleep, bleep, click, 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 bleep, and so on. And we tell them that we want them to listen out for the bleeps. The electrical response to a click will be different from the electrical response to a bleep. The electrical response to a click will be associated with a flat line at about 300 milliseconds, a third of a second after the stimulus. But the electrical response to the sound that they're listening to, the bleep, will be associated with a big waveform at about 300 milliseconds, which we call the oddball response, because it's an oddball stimulus. It's the unusual stimulus that we've asked them to listen out for. And that's the first time we see a difference in the auditory evoked response based on cognition, based on them listening out for one particular sound. So what does that mean? Well, basically, it means that if you're listening to an auditory stimulus, it takes you a third of a second to work out what that stimulus means. So your auditory consciousness is following reality at about a third of a second all the time, not able to catch up. And obviously all the other senses are doing that as well. But the processing for different senses takes a different amount of time to get to those conscious centres and to work out what's going on. And yet we perceive reality as a single continuum. Yep, we live in this world, we perceive it as a real world, we perceive it happening right this moment, and we perceive it all happening together. Let me give you an example of that. Let's imagine that you're walking down a street in a rough part of town one evening, let's say Deepestockifitu, I don't know, okay? And somebody throws a brick at you. And that night you're in the pub, and you say to your friend, I was walking down the street this evening and some bastard threw a brick at me. And that's how you remember it, yeah? That's your conscious memory of what happened. But the way you put that information together isn't like that. You go, something moving, it's coming towards me. It was a brick, you bastard. Okay? It's a different order of sensory stimulation. What's happening is, when that sensory information from sight, hearing, touch if the brick hits you, gets into the central nervous system for the first time, it's given a timestamp. And when you finally worked out what's going on and worked out what it all means, it's still got its timestamp, so you know when it arrived. And your cerebral cortex, which is desperate to make this view of the world that has you as a single entity, puts it all together with its timestamp and arranges the world as it chooses to see the world. But that's not how your senses perceive the world. It's all different. So the added value, the information that you're putting together, is what goes to perceive your conscious experience of the world. And your conscious experience of the world is not your sensory experience of the world. It's the sensory experience that you have filtered through this big imagination machine that is your left cortex. So, where does information go when it gets from the brainstem? Well, it's very similar to vision. It goes to the tectospinal tract for postural responses, head and ear positioning. I hear a sound. It goes to the reticular formation for alerting input. Ooh, I hear a sound. And it goes to the auditory cortex for conscious hearing. What does that sound mean in terms of language, in terms of threat, in terms of potential for food, all those things that are important. I want to look now at a particular segment of the added value processing system for sound. And the reason I want to look at this particular segment is, one, it's relatively simple to work out how it works and what's going on. And second, we know the neurons that can do this. We know where they are in the brainstem, we know how they're wired up, and they're in the olivary nucleus. So it's a fairly basic, early processing that goes on. And it's the auditory processing that tells us whereabouts in space a sound is coming from. So if you're sitting listening to something and it's coming from a particular point in space, I don't know, me droning on about hearing, then your hearing system is able to pick up whereabouts in space that sound is coming from. And these are the cells that do it. They are the signal delay spatial processing network. Now we know that the transmission of sound through air is happening at a finite velocity. Yep, speed of sound, about 330 meters a second or something of that order. So for sounds that come from the side, they arrive, if they come from this side, they arrive at this ear before they get to this ear. Okay? And so there is a delay in processing those sounds from this ear and from this ear. But we have centers within the brainstem that can compare those sounds and say, no, those two sounds came at different times, but actually they're very similar. They're the same sound. They're just coming from a different point in space, and that's why we hear them at different times. And then those sounds are put into this spatial delay network. And from the right ear, they go round this loop and feed off to these processing neurons. And from the left ear, they go through this loop. And we know again that there is a finite speed of action potential velocity along a neuron. So, if the sound comes from the middle, right in front or right behind, it's going to get to this cell from the right ear first and from the left ear first. It's going to get to this cell at the same time from both ears, because the pathway to that cell is the same length from both sides. And it's going to get to the cell at the top from the left ear before it gets from the right ear. If those cells are arranged so that they only generate an action potential when they're stimulated from both pathways at the same time, then for a sound that comes directly from the center, only this cell is going to generate an action potential, because only that cell receives both inputs at the same time. So an action potential from this cell means it came from in front of you. If it comes from the right side, it's going to get into the right ear before the left ear. And so if it gets to this side first and goes around a shorter pathway, it's going to get to this neuron way before the signal from this side. But conversely, because the pathway to the top neuron is shorter from the left from the right, a sound that starts off here earlier is going to get to that neuron at the same time as a sound from the other ear. So when a stimulus from each pathway gets to that top neuron at the same time, we know it came from the right field, because it must have got to the right ear before the left ear in order to do that. So we've generated an array of neurons that respond to sounds that come from particular places in the auditory field. The point is that we are adding value to the sounds that we're getting, because now when we go past the olivary nucleus, we can say this is a sound and it came from over there. The next thing we add to it is movement. This is a sound and it came from over there and it's moving that way, or moving that way, or not moving. And different neural arrangements code for extracting that different information from the sound signal. Back to Hoki. Hokie suffers from congenital deafness. It's a genetic condition. And now that we've gone through sound and talked about sound, I can give you more details about it and also tell you what we're doing to try and not treat this condition, because it doesn't have an easy treatment in dogs. It's a cochlear implant job, and dogs are not really high on the list for cochlear implants at the moment. But to reduce the incidence of the disease in the population. The problem with this disease is that it's not a simple genetic disorder. It's not Mendelian. So the genes which code for it are complex and difficult to predict. And we know, for example, that in the Dalmatian population, for example, the incidence of congenital deafness in New Zealand at the moment is, we think, about 4%. It's really hard to get information from breeders, because all the breeders want the people that are going to buy their puppies to know that they've got no deafness in their line. So they're not going to tell you about their deafness. But looking at the data that we've been collecting for the last 5 or 6 years, it's looking like around about 3, maybe 4%. We know that if we breed 2 deaf dogs together, there is an incidence of deafness in the progeny of around about 30 or 40 or even 50%. So we're not going to do that. The problem is that dogs can be affected bilaterally and be deaf, or they can be affected unilaterally. If they are, and heterozygous is the wrong word for this, but if they have one expression of those set of genes that gives hearing and one expression that doesn't, and if the stem cells for the cochlea come from one group of cells on one side that don't hear and a group of cells on the other side that do hear, then they're unilaterally affected. They hear with one ear and not the other. But it's very, very, very difficult to tell when a dog is unilaterally affected, because Dalmatians, despite appearances to the contrary, are quite bright dogs and quite intelligent. They can work things out. And often their owners will not notice that they are unilaterally deaf, because they'll be picking up other cues, you know. When I shout him in the park, he comes to me. Of course he does. He hears the sound, he associates it with the owner, he looks around for the owner, and he runs off to the owner. And the owner thinks, oh, dog heard me, looked at me, came running towards me. So it's very difficult not to breed from animals that are unilaterally affected. And that means that it's very difficult to get the incidence of deafness in the population below this simmering 4% or thereabouts. Very easy to spot deaf puppies and to remove them from the population, but many breeding dogs for many years have been unilaterally affected, it seems. But we can now develop a test which can detect unilaterally affected animals. We can provide a click stimulus to one ear and then the other ear, and we can record the electrical activity of the brainstem from those puppies whilst playing a click stimulus to just one ear or just the other ear, and see what the response is. So, this is the brainstem auditory evoked response, the BAER, sometimes called the brainstem auditory evoked potential, the BAEP. So don't get confused if you read it differently. But this is the BAER of a normal hearing dog. The big spike at the start here is the electrical activity that you get from the click stimulus. Because again, you're putting a speaker here by the animal's ear. It's got an electrical coil in it. You're stimulating it, and it's inducing a voltage change in the electrodes, which is bigger than the voltage change the electrodes are recording from the neural centers. But importantly, following that click stimulus, you can see these characteristic waveform responses that appear in the signal. And so if we play that signal to just one ear, and this is really technical, we stuff the other ear full of cotton wool before we start, and we put the speaker by this ear and we play it fairly quiet, then if that ear hears, we will see that waveform response coming from the brainstem. But if the ear doesn't hear, this is the response we see. We've still got that stimulus artifact, the spike due to the electrical activity in the speaker, but following that we've got a flat line, no response. And so we can now say to breeders, this dog doesn't hear in one ear, and we can tell them which ear. And they will say, oh no, it's the best dog I've bred for 10 years and I really wanted him to be in the shows and so on. And we say, do you want the incidence of deafness in the breed to reduce or not? And then we have this full and frank discussion and so on. But usually that means that they won't breed from unilaterally affected dogs. We haven't yet got data for New Zealand to say how that's affecting deafness in the population, but there is data from Holland that says that if the testing is done rigorously on all breeding animals, and if all unilaterally affected animals are removed from the breeding population and they just get castrated or neutered and homed as pets or whatever, if those unilaterally affected animals are removed from the breeding population, then the incidence in Holland in 10 years dropped from 8% to 1%. So it's not solving the problem, but it is removing the problem over several generations by not breeding animals which are predisposed to develop that particular problem. And we're doing that because of a neurophysiological test that can tell us definitively whether animals can hear in one ear, both ears, no ears.